retina, right? Now let's see the central neurophysiology of vision. So, if we close our right eyes, I don't know if you hear my voice when I'm talking. It's okay now? Okay. So, if you close your right eye and only look through the left eye, then the world will be seen not in a circular way, but in this manner, right? So, part of the circle cannot be seen. How can we test this? For example, I close my right eye, okay, I'm looking at the center. Now, when I keep looking the center, and I also pay attention to this, I can see that, right? When I move it upward and go here, I cannot see that. So this means this part of the visual field is blind, so I cannot see that part. If I keep my right eye, right, and move this downward, I can see this, but now I cannot see that. So this means from this point below, I cannot see anything, right? I can do the same thing when I move this way. Okay, now I cannot see that. So this means from this point that way, I cannot see anything. So we can do this. We call this perimetry. Perimetry. So we can do this. We can keep our look to the central part, right? This way we can see or we can define the field of vision for left eye. So the field of vision for the left eye is like this. Central part, lateral part, medial part, or central part, temporal part, nasal part. Got this? Now, we can do the same for the right eye. So, we keep the left eye closed, then do the perimetry for the right eye. So we'll see something like this, right? So now we can put the visual field of the right eye and visual field of the left eye overlaid, right? We'll see the visual field like this. So the central region is covered by the fovea. Then we have right visual field, left visual field. This region is called binocular visual field. Why? Because this is part of the right visual field and part of the left visual field. So we can see this region by both eyes. But this region and this region is called monocular visual field because only one eye can see this, the right eye. And only the left eye can see this part. So we call this monocular visual field. So we have a visual field. Some part is monocular. The central region is binocular. The central point in the binocular region is called foveal region. Right?
Why don't see the nose?
Why don't see the nose? So the nose causes our right eye not be able to see everything on the left side. Only see part of the visual field. And this node causes the left eye to be able only see a little of the opposite field. So this is the effect of nose, but also the effect of pupil, right? Pupil is also small, and it doesn't allow light from each part of the visual field to come into the eyes, right? So now we can see an eye and the visual scene in front. So there is a letter F here, and we have some quadrants with different colors, right? So again, we can do perimetry for the right eye, for the left eye. So you can see that the letter F is located in the middle part, in the central part, right? Then we can see the whole part of the visual field on the same side and a smaller part from the opposite side, right? And in the left eye, we can see the whole part of the left visual field and some part of the right visual field, right? So it's the same actually. So most of the ipsilateral field, a little of the contralateral field. Most of the ipsilateral field, a little of contralateral field. When we put them overlaid, we have binocular field, monocular field, right? We can put these two together and see this, right? So the letter F is located in the central point. Then we have right visual field, left visual field, binocular visual field. Got this? So this happens in the outside of the eye, in the visual field. Now let's see what happens in the retina, right? So right visual field, left visual field. Right retina, left retina. So you can see that in the retina, the figure is inverted and reversed. So the F is inverted, and you can see the location of this purple goes from down and left to top and right. Right? The same also happens here. The purple is down and to the left. Now it's top and to the right. So the image, the visual field that goes to the retina is inverted and reversed. Clear? Okay. Now, this is the lateral side of the visual field. Lateral side. Medial side. Medial side. Visual field. But what about retina? This part of the visual field can be seen by temporal retina. But the lateral side of the visual field is seen by nasal retina. So on each side, the nasal half sees lateral part. Temporal retina sees medial side. And for this eye, nasal half sees lateral part, temporal retina sees medial part. Got this? Now, what happens if we want to have only one image? So now we have two images. One image on the right retina, another image on the left retina. But we should have only one image, right? So you can see that temporal retina of this eye sees exactly the same part of the image that sees by nasal retina of the opposite eye. So, temporal image of each side equals nasal image of the opposite side. Again, temporal image of this eye equals nasal image of opposite eye. Right? So if we want to have one image, it means this part and this part should go to a point in the brain, to the same point. And this part and this part should go to another region in the brain. Right? So then half of the image is in one hemisphere, another half is in another hemisphere. Got this?
There's difference between, for example, temporal side of the retina and temporal side of the visual field.
Yes. So temporal retina on each eye can see the same part of the image that is seen by nasal retina of the opposite eye. So their image is almost the same.
For example, when I see the door, see by this temporal side and this nasal.
No, not the door. When you look at me, I'm on your fovea. Right? Now, your nasal part of this eye sees that part. Temporal half of the other eye also sees that part. Right? And temporal part of this eye and nasal part of that eye see this side. When you look at me, when you keep your eyes fixed, right? So if you move your eyes, then the central region also moves. Okay? So, but we should keep it still and don't move it. So, again, temporal part of this retina, nasal part of this retina have the same image. So they should go to the same hemisphere. But nasal part of this retina, temporal part of this retina have almost the same image, so they should go to another hemisphere. Let's see. Left eye, right eye. Temporal retina, nasal retina. Temporal retina of the right, nasal retina of the left. They have the same figure. So they should go to this hemisphere. But now, nasal part of right eye, temporal part of left eye. They have the same image, right? So they should go to this hemisphere. So, how this is possible? So, temporal region on each eye go to ipsilateral hemisphere. Right? But nasal part of each retina goes to the contralateral hemisphere. Right? So, from the retina to the visual cortex, we have retina, optic nerve, optic chiasm, optic tract, lateral geniculate nucleus of the thalamus, optic radiation, visual cortex. So this pathway is called new visual pathway. New visual pathway. This is new pathway. So the new system. Right? Got this? So it's like this. Okay? So temporal, nasal. They go to this side. Temporal, nasal. They go to this. Right? Temporal of this side, nasal of that side. Got this? Okay. They go to the cortex. Now, let's see separation of the superior retina and inferior retina. I talked about nasal temporal, but what about superior, inferior? Right? So let's see. From the lateral geniculate nucleus of the thalamus, some fibers go to the superior part of the visual cortex. You know that visual cortex is located on the medial side of the brain, right? Medial side, around the calcarine fissure. So above the calcarine fissure, we have cuneus or cuneus. Below the calcarine sulcus, we have lingual gyrus, right? So some of the fibers go to cuneus, but some other fibers go to lingual gyrus. So those that go to the superior part go through the parietal lobe, and fibers that go to the inferior part turn around the ventricle and go through the temporal lobe. So optic radiation has two parts, parietal part, temporal part. This inferior one is called Mayer's loop. Got this? No, only primary visual cortex. Now I'm talking about primary visual cortex, right? So now you can see here, from superior part of the retina, the green fibers go to the LGN thalamus. From here, they go to the superior part of the visual cortex. So this pathway. But from the inferior part of the retina, fibers go to LGN. Then from LGN, they go through the loop of Mayer to the inferior part of the visual cortex. Right? So, from the fovea, fibers go to the LGN. Then from LGN, they go to both superior and inferior part. Got this? Now, superior retina sees inferior part of the visual field. Inferior retina sees upper part of the visual field. So the image that comes to the retina is inverted and reversed. The same figure goes to cortex. So the image, the cortex is also inverted and reversed. So from inferior visual field, information go to superior visual cortex. Superior visual field goes to inferior visual cortex. Got this? Okay, now, most of the fibers that originate from the fovea go to this part of the visual cortex. The larger part, yeah. But peripheral part of the retina is represented gradually into the anterior part, right? And the size of, actually this is not clear here, but we can see here. So we can see that from a small region of the fovea, fibers go to a large area of the visual field, but from a very large part of the retina, fibers go to a very small region of the visual cortex. So that's why acuity of the vision is higher for the foveal region, right? So we call this primary visual cortex. Around the primary visual cortex, we have secondary visual cortex, which is related to higher level processing of visual information. Now, let's see what happens if we look at an object and how it is conveyed to the visual cortex, right? So we have an arrow here in front of the two eyes. So the tail is here, the tip is here. So when we look at the center hits the fovea. So the fovea of both eyes can see this part of the image. Now, let's see the right eye. what happens in the right eye? So the nasal part sees the lateral side. So from here to here is seen by nasal retina.
Of the right eye on the temporal side of the left eye.
Yeah, temporal side of the right eye sees from here to here. So part of the binocular visual field. So you can see that temporal part of the retina can see from the central point to here. Right? So the tip is not seen here. But now let's see the image in the left eye. So the central part is seen by fovea. The nasal retina can see the main part, the tip to here, and temporal retina can see part of the contralateral image, this way. But this one is not seen here, right? So from here to here and from here to here are the same part of the image, and from here to here and here to here are from the same side. So that's why temporal part and nasal part of the opposite side go to the same hemisphere, right? And nasal part of the opposite eye, temporal part of the same eye go to this hemisphere. So half of this image is seen in this hemisphere and another half is seen in the opposite hemisphere. But remember, the connection is contralateral. The tip is on the left, but it is seen on the right side. The tail is on the right, but it is seen by left hemisphere. Got this? Good. Now let's see what happens if we have damage in the visual pathway. So, imagine that there is a damage in the optic nerve on the right side. What happens? So, temporal retina, nasal retina are lost. So, the total right eye will be blind. So the visual field related to the right eye is not seen. Visual field of the right side is blind. Right? So this part, so consider that this one is part of the visual field. Visual field. Right? So this part of the visual field is seen by temporal retina. This part is seen by nasal retina. So the visual field of the right eye is totally blind. But when we do perimetry, we put the figures overlaid. So if we put these two together, then we'll have something like this. So we want to move this here, right? So this point corresponds to this, right? So we'll see something like this. Okay? So this part will be totally blind, right? But because the left eye is intact, it can see this part. But if we close the left eye, then the right eye is totally blind.
Why this case not effect on the left visual field?
Why? Left visual field? You mean this? Because this is for the left eye, and the left eye is intact. So, if we keep the left eye closed, then by the right eye, the person cannot see anything. Because the optic nerve for the right side is totally cut. Right? Does it make sense? Okay. So now, yeah.
What happened if the right hemisphere was cut?
Hemisphere, I will talk. Okay, let's see, little by little. Okay? So, this is damage in the optic nerve. Now, let's see if we have a damage in the optic chiasm. So, in the optic chiasm, we have only nasal fibers. So, nasal fibers from here go through the optic chiasm that way. Nasal fibers of the left eye come through the optic chiasm to the right side. They are cut now. So the nasal retina sees this part of the image, right? This nasal part sees this part of the image. So the medial side is intact. Lateral side on both sides will be blind. So you can see that the lateral side on both sides are blind, but medial part is intact. If there is damage in the optic chiasm. What else? If we have a damage in the optic tract. So in the optic tract, we have fibers from the temporal retina of the same side, nasal retina of the opposite side. Right? Temporal retina of this side sees this part of the image. Right? Nasal retina of the opposite side sees this part of the image. Right? So, the medial part of the right visual field, lateral part of the left visual field. Lateral part of the left visual field, medial part of the right visual field. So, they will be blind. Got this? Now, let's see what happens if we have a damage in the loop of Meyer. So I told you that it's located in the inferior part, right? It receives fibers from the inferior part of the retina, but it sees superior part, right? So, it should be similar to C when we consider the side, right? But because only inferior part is damaged, then superior part of the field is blind. Inferior part is intact because we have other fibers that bring the information to the cortex. Okay? Now let's see what happens if we have damage in the visual cortex. Your question. Right? So it's similar to C. Because temporal fibers of the same eye, nasal fibers of the opposite eye come here. So the block that happens here is similar to this one. So we have the same figure here. But the only difference is that we have a region for fovea here. The reason for that is not exactly known, but maybe because fibers from the fovea goes to both sides and also to superior and inferior part. So that's why when we have a damage in the visual cortex, fibers from the other eye can compensate for this lack. Okay? So we have a patient that cannot see the lateral side of the image on both sides. Lateral part on both sides. Optic, quiescent. Yeah, we have a patient which is blind on the lateral side of both sides. Optic tract. So lateral part on both sides. So optic quiescent. Right? So we have a patient that in a perimetry test has a problem or is blind on the left side, both for right eye and left eye. So the the part of the blindness is on the left side. Yeah, left side. Optic tract. Which side? Right side. All right? Got this? Okay. Okay. Now, I told you that in the new visual system, fibers from the retina go to the thalamus. In the thalamus, we have 6 different layers. Please pay attention to me. Please pay attention to me. You also. You. Okay. In the LGN, we have 6 different layers. Layer 1 through 6. Right? So I told you that from temporal part of the retina on the same eye and nasal part of the opposite eye, fibers come to the lateral geniculate body of each side, right? So those that originate from temporal retina come to the ipsilateral LGN and they go to layer 2, layer 3, and layer 5. So 2, 3, 5. Right? 2, 3, 5. From where? Temporal part of the same retina. Right? But from nasal part of the opposite eye, fibers come to layer 1, 4, 6. Got this? The same also happens for the opposite side. Okay, so from ipsilateral eye, they go to layer 2, 3, 5. From contralateral eye, they go to 1, 4, 6. But remember, in our previous session, I told you that we have P cells and M cells. P cells in the retina, M cells also in the retina. M means magnocellular, P means parvocellular. Yeah, so in the LGN, layer 1 and 2 are macrocellular. But layer 3, 4, 5, 6 are parvocellular. So they have small cells, but these two have large cells. So from M cells of the retina, fibers go to layers 1 and 2. From P cells of the retina, fibers go to layers 3, 4, 5, 6. Now again, M cells of the temporal part of the same retina go to layer 2 or 1, 2. Same side. I told you 2, 3, 5 from the same side, 1, 4, 6 from opposite side. Right? So from the same side, M cells come to layer 2. From the opposite side, M cells go to layer 1. So from the same temporal retina, same side, P cells go to layer 3 and 5. From contralateral nasal retina, P cells go to layer 4 and 6. Very good. So, both images that come from different eyes are separated. And images that come from M cells and P cells are also separated in the LGN. Right? Then from LGN they go to visual cortex. The same also happens in the visual cortex. So, fibers from M cells go to LGN. Which layers? Layer 1 and 2, right? Ipsilateral go to 2, contralateral goes to 1. Okay. From here, magnocellular, they go to layer 4 of the cortex. Which subregion? C alpha. So we have A, B, C alpha, C beta in layer 4. Right? But fibers that go and originate from P cells of the retina go to layer 3, 4, 5, 6. Then from here, they go to layer A of layer 4 and C beta of layer 4. So they remain separated. Got this? M cells were related to movement and black and white stimulation and direction and orientation. Remember that? But P cells were related to color and details. So information about color and details go to these layers, but information about movement and orientation go to this layer.
What's it mean by detail?
Detail? I'll talk about this. Okay? Detail means every feature that we can see in an object. For example, you may have a bird here, right? So it can be a dove or a parrot. You know parrot? Parrot and dove. Right? So, how can we actually tell the difference between these two? Because of the details of the image. Right? So they are different. The shape, the form, the appearance, the color, everything is different. So these are the details of the image. So again, you can also see the details of this image. For example, this one shows this way, but here you have two, they are different actually. So the details of the image is different. So these details can be differentiated by P cells. Got this? Okay. So from here, information goes to all over the cortex. I mean, in all layers of the cortex. From here, they go downward and upward, and some collaterals go to some column-like structures called color blobs. And they are important for detection of colors. So this pathway is important for color and detail, but this pathway is important for black and white and fast movement. Got this? C alpha. Yeah, first they enter here, but after that they spread all over the cortex. Yeah. Then they go to different layers. Right. Okay. So now the information reached the cerebral cortex, area 17, around the calcarine fissure. Right? So here we have different type of cells. So remember from our previous session that ganglion cells had on-center or off-center receptive fields. Right? Some of the cells in layer 4 of the cortex have the same feature. So the central region is excitatory, Peripheral region is inhibitory. So if the light illuminates the center, the cell is activated. If the light illuminates the surround, the cell is inhibited. So we call this concentric receptive field. So these cells are only found in layer 4. Yeah, it can be on center or off center. So it can be like this. So these are off center, right? So surround is excitatory. So center, surround. Right? So cells here are activated by a light that happens in the center or in the surround. So we call them concentric receptive fields. Another type of cell is simple cell. So the receptive field is like this. It has a central region which is excitatory and inhibitory surrounds like this. And they are sensitive to orientation of the bar of the light. So this one is a border or a bar of light, right? So the orientation is this way. So the cell is only activated if the bar of light happens in the center and with this orientation. If the orientation is like this, the cell is not activated. If the orientation is this, is not activated. This, not activated. So only is activated when the bar is like this. And if you put the bar here, the cell is inhibited. If you put this here, the cell is inhibited. Only here and in this way, the cell is activated. Right? So we may have another simple cell with a receptive field like this. So if the bar of light has this orientation and happens in the central region, the cell is activated. If the bar is here, inhibits the cell, the light here inhibits the cell. If the orientation is like this, again, the cell is not activated. So we have some simple cells that are activated if there is a bar of light with this orientation, other cells by this orientation, other cells by this orientation, other cells by this orientation. So for every orientation, we may have some cells. So now, let's see why these are important for us. So bar of light means the border of an image. So I told you before, we have a border here, maybe a border here, a border here, which is vertical, right? I'm here, and my hand is like this, so we have a border like this. This is in my hand, so it has another border. So some simple cells are activated by this one. Other simple cells in your brain are activated by this. Other simple cells are activated by this. Other simple cells are activated by this. Others by this. So in this way, simple cells in the visual cortex are activated by different lines in this figure. So you can see this figure, you can see several borders, several edges, and each of these edges activates some of the cells in your visual cortex. Right? So for this type of cell, we need two features, orientation and position. Position should be in the center, orientation should be specific for this cell. Right? So now we have another type of cell called complex. The receptive field is similar, so this one also is activated by the same orientation, but it is not position specific. So you can move the bar here or here or here or here. So all of them can activate the cell. So the cell is more complex. Why? It responds to more features. Not only to the orientation, but it is not important for this cell where in the receptive field this bar is located. So in any position it is activated. Right? So we may have another complex cell which is activated when the bar orientation is like this. So here, 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 or here, the cell is activated. So it's different from simple cells. Wait, please. Now we have hypercomplex. Hypercomplex means the cell is activated by an orientation which is not important or which is not specific for position. So you can move it this way or this way. But the length of this bar is important. If the length is so long, the cell doesn't respond. Only to this part or this length, it is reactive. So we can say that we may have the same borders, but one is short, the other one is long. So some of cells only respond to the shorter one. Some other cells only respond to the longer one. Right? So we have 4 different types of cells in the primary visual cortex, and they can analyze different features of the image.
And the second type, the changes in the orientation is not active, but if the change in the position is
Inhibited. Yes.
But in the complex and hypercomplex, that's
Position is not important. So whenever the orientation is okay, the cell is activated everywhere in the receptive field. But here length is important, right? So if the length goes out of this, the cell is not activated. Yeah. Yeah, it doesn't have inhibitory area. So, continue. So based on this, so we have simple cells in the V1, we have complex cells, we have hypercomplex. So hypercomplex cells may be specific for length or specific for some shapes or specific for other characteristics, right? So we may have some hypercomplex cells that are activated by this angle. So when 2 borders come close to each other and make an angle, this cell is activated. But if the angle is wider, the cell is not activated. Right? So we have a mixture of cells. Each of them are specifically activated by some features. So in the secondary visual cortex, the cells are more complex. For example, some of them are activated by a circle, but they are not activated by a square. Right? Some other cells can be activated by a triangle, but not by a triangle which is reversed. Maybe. Right? So, for any type of figure, we have some specific cells. When we go out of the primary visual cortex and we go step by step in the secondary visual cortex, the cell becomes more and more complex. Right? So now, let's see what happens if we remove V1, primary visual cortex, the person will be blind. We have seen that. Right? If you remove the cortex, part of the lateral side and medial side of this eye will be blind. But the person is still able to detect light intensity, movement, and gross pattern. This may happen because of the activation of superior colliculus. But we have also secondary visual area. So in the secondary visual area, we have V2, V3, V3A, V7, LO. So we have several areas, right? So collectively, we can say that we have 2 pathways for analysis of the signal. So they originate from V1. Some of the processing goes this way to the temporal cortex. Other pathway go dorsally, go to the parietal cortex. So P cells that are related to color and details. So the information from P cells is analyzed in the ventral stream, but information from M cells is analyzed in dorsal stream.
I activate to the function of the brain.
Yeah. Yes. So temporal part is important for color and detail. Parietal part is important for movement, location, form, speed. Yeah. And now, remember from neurophysiology that I told you, if there is damage here, medioventral temporal region, what happens? The object. We call that visual object agnosia. Right? So why? Because the details of the image cannot be analyzed. So the person is not able to differentiate between a key and a pen. Cannot recognize them. Right? So agnosia happens here. And this processing goes ahead. In the inferior side, it can process the face. Face is a more complex feature. So damage here causes prosopagnosia. Remember that? Prosopagnosia. The person is not able to recognize familiar faces. Now, this pathway. If there is damage here, the person will have a problem understanding space. Yeah, so the person usually ignores the left side of the visual field. So we call this hemisensory or hemispatial neglect. Right? So information from M cells that are related to the space, motion, and form are analyzed here. So when there is interruption here, they cannot be analyzed. Got this? Okay, I told you that we have new pathway and old pathway. The old pathway goes to different areas. So from retina, some of the fibers go to suprachiasmatic nucleus, internal clock, circadian rhythm. Good, very good. Some other fibers go to pretectal region. Pretectal nuclei are important for pupillary light reflex. Yes. So, meiosis and mydriasis, right? Some other fibers go to superior colliculus. They are important for movement of the eyes. So, in the new pathway, fibers come to LGN. From LGN, they go to cortex. This pathway is for vision. In the old pathway, fibers go to suprachiasmatic nucleus, pretectal area, superior colliculus. Right? So they are for circadian rhythm, for pupillary light reflex, and for eye movement. So they are separated. Now let's see some of the functions of this old pathway. So pupillary light reflex. When you shed light into the eye, the retina is activated. So it goes through nerve number 2 to the pretectal region. Then from pretectal region, fibers go to Edinger-Westphal, which is the origin of the preganglionic parasympathetic fibers. Right? Preganglionic parasympathetic fibers go to ciliary ganglion, make connections with postganglionic parasympathetic fibers. They go to the iris muscle, circular one. The circular iris muscle contracts, pupil decreases. Pupil will be smaller. So in response to light, we'll have meiosis. Right? Sympathetic innervation causes mydriasis. Yeah.
The person died
Why that was the first thing they check the eye?
Because,
The eye.
yeah, because the centers related to pupillary eye reflex are located in the brainstem, right? And the brainstem is a vital region of the brain. If there is damage here, probably the person will die. Okay? So when there is no response to the light, it means there is some damage in the brainstem. Right? So that's why we can be actually more sure about the death of the person. But if there is light reflex, what does it show? It means brainstem is intact. It is still intact. It is working. Right? So the person is alive. The person is still alive when we can see the reflex. Right? So, extraocular muscles, you know them.
Yeah.
Superior rectus, inferior rectus, lateral rectus, medial rectus. Superior oblique, inferior oblique. So, lateral rectus moves the eye this way, laterally. Superior rectus elevates. Inferior rectus depresses. Medial rectus adduct. Medial side. Superior oblique, adduction and depression. Inferior oblique, adduction and elevation.
Which muscle?
Superior oblique, inferior oblique.
The downward.
Yeah. Superior oblique moves the eye downward. And also adduction. So when the eye is adducted, superior oblique bring it down. Right? Did you test it? So based on this, we can have different type of eye movements. So we can have